Thanks very much, Finn. And yeah, welcome everybody to this our webinar. And this forms part of the UK Freshwater Quality Research Programme. And I know many of you have joined these before, but just in case we know there's a few new people, this programme is funded by UKRI, by the Natural Environment Research Council and by DEFRA. And the aims of the programme is really to understand the sources and behaviour of pollutants within river systems, increase our knowledge of how pollutants impact freshwater ecosystems, look at where mitigating pollution load into river systems, and ensure that the outputs from these research projects inform policy changes and useful for the regulatory organisations, many of which I know are listening today. So great to have you all here. And the programme funded 5 research projects. And over the last few months, we've been hearing the progress of each of these projects. And this month's turn is Pacific. And so Dan Read is here from UKCEH to tell us a little bit about how they're progressing. So we'll pass to him shortly. But you can catch up on some of the other webinars via our website. And we'll be hearing, thanks, Finn. So these are ways that you can contact us through our website, where you can catch up with other webinars that we've had. You can email us at freshwater@leeds.ac.uk, and you can follow us on Twitter for information and comms. And as I said, we've heard from some of these projects, and our upcoming webinars for the next 2 months will complete this year's summary or updates from the project. So the next one next month will be Quantum, that's looking at livestock, particularly livestock farming on water quality. And then lastly, in June, we'll be hearing from LTLSFE. So thanks for joining. And I'll pass over to Dan, who will tell us much more about Pacific and the project he's leading.
Okay, thank you, Pippa. I'll just share my screen, and can you just confirm that's in full screen for you?
Yeah, that's great.
Brilliant.
Thanks, Dan.
Okay, thank you. Okay, well, thank you for the introduction, Pippa, and good morning, everyone. So as Pippa said, I'm going to give you a quick run through of the Pacific project, and just to give you a very brief outline of how I'm going to structure this presentation. Next slide. So I'm going to spend the first 10 minutes or so just introducing the wider topic of freshwater ecosystem health, and why we're interested in doing this research, really. Put some of the work we're planning to do in the context of historical trends and looking at the sort of current status of freshwater ecosystems as well as future threats. I'm going to then spend a little bit of time focusing on the sort of main focal point of the project, which is the microbes within freshwaters, how we can use those as indicators of freshwater ecosystem health, then run through an overview of the Pacific project and just talk a little bit about some of the data analysis and early results. So I'm sure, I think I'm right in saying that all of you on this call have an interest in freshwater ecosystem health, and it won't have escaped your notice that this is a topic that's had a lot of media attention recently, both within the conventional press and social media. I've selected a few headlines here, and these are just from the last 12 months, highlighting some of the sort of media attention around freshwaters within the UK. And these broadly fall into 3 main categories. The sort of reporting on the Water Framework Directive data, so this figure that often gets thrown around 14% of rivers in England having good ecological status, the release of untreated sewage into rivers from combined sewer overflows and the sort of focus on the water industry around that. And then also linked to this is the microbiological safety of rivers from a recreational or bathing water perspective. And I think from this, you could be forgiven for thinking our rivers are in an absolutely terrible state. And I've seen, it reported that, rivers are the water quality is declining and things are getting worse. But the reality is that actually our rivers are in better status than they have been for some time. So these are some figures from a paper that was recently published by some of my colleagues at UKCEH. This was from a NERC-funded project called ChemPop. And what my colleague Andrew Johnson and his collaborators did was look at an analysis of over 200,000 records of macroinvertebrate communities that live within freshwater. And this spanned about a 30-year period from 1989 to 2018. And these results generally show patterns of improving diversity and improving community composition of these freshwater indicators. So in the top left-hand side, we have the family richness. The two lines represent the full database of 200,000, over 200,000 records in blue, and then the orange is a subset of sites that have better temporal records. And you can see that there was quite significant improvements in the 1990s and possibly a leveling off since the sort of early 2000s. Focusing on taxa, the EPT taxa, so these are mayflies, stoneflies, and caddisflies. They are particularly sensitive to pollution. You can see there's been a sort of sustained increase in the diversity of these taxa that are recorded on average across England. So this is Environment Agency data. And the bottom two charts just show indices that the Environment Agency used to measure ecosystem health, and again, showing similar trends. Now, the reason for this actually is that water quality on the whole has got better in England. And this is largely driven by the introduction of the Urban Wastewater Directive, so some EU legislation that regulated on the emissions from wastewater treatment works. So this is a similar sized data set. This spans from 1986 to 2018. And you can see for biological oxygen demand that there's been a decline. So this is a measure of organic pollution, sort of gross organic pollution, generally quite a good marker of poorly treated wastewater. That on average has declined across English rivers. Ammonium, metals such as copper and zinc with a less convincing decline or more noisy trend, phosphate as well. The only determinant that actually sort of bucks this trend is nitrate, and that's because it's largely a legacy pollutant that can persist for a long time within the system, within groundwater and in the subsurface. So it takes a long time to work its way through and into freshwaters. So based on this, you could argue that actually freshwaters are in a better state than they have been for some time, certainly within the last 30 years. But I would argue that there are still some significant problems that we need to address. And the first of these is that these are just average trends. So within this, there are sites that will have definitely got worse. There are locations within rivers that will have got worse, and some will have stayed the same, even if we have this general background trend of improvement. But secondly, we can't assume that things are going to stay the same into the future. So this is a very nice paper that came out, I think just a couple of weeks ago from the University of Sheffield. And what they did was look at various different climate scenarios, these RCP climate scenarios. So each of these graphs represents a group of macroinvertebrates, and simplistically on the sort of left-hand side of each graph, you have the low impact climate scenario, and on the right-hand side, you have a higher impact climate scenario. So generally higher levels of warming. And what they showed very nicely, actually using the same Environment Agency data, that even if everything else was to stay the same, climate warming is likely to shift the composition of macroinvertebrate communities to different community compositions. And then actually, more importantly, they showed by comparing this to published ecotoxicological data that these communities are likely to shift to ones that are more sensitive to chemical pollutants. So as I said, even if we were to keep everything the same, we're likely to see shifts in community composition that make these more vulnerable to pollution in future. The final point is that actually the nature of pollutants that we see in freshwaters has changed over time and has become much more complex. So an example of this is these emerging contaminants or emerging pollutants. You can see here on the left-hand side just some examples of these sort of the nature of types of pollutants that we can frequently measure within freshwaters. So these include things we use within our household, so pharmaceutical and personal care products, the medicines we take, anti-inflammatories, antibiotics, things we use around the household, soap, shampoos, cosmetics, industrial chemicals that make their way into household products and food, so sweeteners, plasticizers, chemical household products such as disinfectants, flame retardants, insect repellents, emerging particulate contaminants, so microplastics, nanoplastics, metal nanoparticles. So the nature of pollution has changed, and actually the diversity of chemicals that we use in our households, on animals, in farming, within industry has really sort of diversified and is likely to continue to do so. And there's this nice paper on the right-hand side that sort of modeled the threat of these organic chemicals to health of freshwater ecosystems. And they showed at a European scale that there are real hotspots of threat, including in the UK, largely driven by levels of wastewater dilution. So these micropollutants, they can make their way into freshwaters by sort of two broad pathways. We have point sources, which is wastewater treatment. So these are things, chemicals we use in our household that we flush down the sink, flush down the toilet. These make their way into wastewater treatment works. Some chemicals can pass through the treatment processes unaffected or not completely degraded, so they make their way out into freshwaters. And also there is a sort of a diversion through combined sewer overflows where they can get through into the environment untreated. Diffuse sources such as agricultural chemicals we apply to the land, so insecticides, fungicides, biocides, herbicides. I would add to that actually road runoff as a major diffuse source of pollutants that make their way into freshwaters. And the real problem with this is that it means we can have mixtures of chemicals, hundreds to thousands of different chemicals that are measurable in freshwaters. And this brings us this huge challenge around understanding which types of chemicals are important and how do they interact with one another. And then to introduce a particular sort of focal point really of the Pacific project is there are classes of chemicals we don't really know much about their ecological impacts. And the one I'm highlighting here is antimicrobials. These are used incredibly widely. We use them as medicines, we use them as household products for cleaning. They're widely used in farming. We spray them on our crops. They're used in industry. And because of their ubiquity, they make their way through into the environment via a number of these different routes. So why might we be concerned about antimicrobial pollution in the environment? Well, freshwater microbes are incredibly important. And so my background is as a freshwater microbiologist, so I'm hopefully going to try and convince you of the importance of this group of organisms. So they are the most diverse component of freshwater ecosystems. You will visibly observe some freshwater microbes, so diatoms, green algae, they're the things that make water go green or produce that sort of brown or green slime on rocks. But that's actually just the sort of tip of the iceberg. There is a huge diversity of organisms, bacteria, archaea, fungi, micro-eukaryotes like protozoa and ciliates, that form a huge diversity of organisms that sort of dwarfs the diversity of other organisms that you might conventionally think about with freshwater biodiversity. But they're not just diverse, they actually play really, really important roles. So they form the biogeochemical engine of the earth and also freshwaters. Their functionality is responsible for turning over organic matter. They're responsible for the cycling of carbon, nitrogen, and phosphorus. They control the metabolism of freshwater, so the production and utilization of oxygen. They play a massive role in greenhouse gas fluxes. So they play this critical role in maintaining the health of freshwater ecosystems. They can also be directly responsible for the health of freshwater ecosystems and the organisms that live in there as pathogens. And as I've already said, they are sensitive to a range of chemicals that we have designed, manufactured, used specifically to target microbial communities outside of a freshwater setting. And these chemicals do make their way into freshwaters. Okay, so how do we go about measuring the health of freshwater microbes? And I've got this fairly simplistic diagram here where we've got a healthy microbiome. This is termed eubiotic or eubiosis. This is a term that's actually came from the sort of understanding of gut microbiomes, where there's a very active line of research about understanding the microbes that live within the human gut and how it relates to the wider health of us. This is an analogy to that, really. We're interested in the microbes that live within rivers and as an indicator of the health of these rivers. So we can have an unbalanced or unhealthy microbial community, a dysbiotic microbial community, maybe caused by exposure to antimicrobials or other chemicals, maybe caused by other stressors such as raised temperature. And we have two main ways in which we can measure the response of these communities or measure the status of these communities. We can look at what species are there, so the composition of species. We can look at the diversity, how many species are there, or we can measure key taxa that might be indicators of a healthy or an unhealthy community. But because these organisms are so functionally important, we can also measure their functional processes. So we can measure the biogeochemical processes they perform, the carbon, nitrogen, and phosphorus cycling. We can measure the physiological traits such as their genome size, their growth rates. And also because microbes are incredibly adaptable and good at responding to chemical exposure. So something you will have heard of probably from another context is antimicrobial resistance, where microbes evolve or gain the ability to resist exposure to chemicals. So there is a huge suite of genes that are used to resist exposure to chemicals that are very relevant for the environment as well. When environments are exposed to these chemicals, it will increase levels of these genes, these antimicrobial resistance genes. And these have the potential to be used as very sensitive indicators for chemical exposure in the environment. So in terms of the methods, we have a number of different approaches we can use. Our sort of primary method is around high throughput DNA sequencing. This is where we can take a sample, we can extract the DNA, and we can effectively decode the mixture of genetic information in that sample. And we can use this to look at what species are in that sample, and we can also use it to look at the functional genetic potential of that community involved with biogeochemical processing, involved with the traits of these organisms, and involved with the way in which they respond to chemical exposure. We also have other methods that we can use to look at directly measure the physiological status of cells, so flow cytometry, or measure things like metabolism or enzyme production using fluorescence assays. But I won't focus so much on that second part today. Okay, so I'm now just going to briefly run through the outline of the Pacific project and sort of introduce the different work packages and how we're approaching this study. So as a quick background, this project started in late 2022. It's going to run until late 2026. It's a collaboration of 4 organisations. So I'm leading the project at the UK Centre for Ecology and Hydrology, working with a group of colleagues who have expertise in molecular microbiology, in the fate of chemicals in the environment, and modelling as well, modelling chemicals. At the University of Bath, the team is led by Barbara Kasprzyk-Horden. Her and her team have expertise in measuring chemical pollutants in the environment. At the University of Oxford, Michelle leads a team who have expertise in multiple stresses and how they interact with freshwater ecosystems. And we have the Environment Agency as a full collaborative partner on our project, led by Kerry Walsh. And Kerry and her team are really interested in the practical applications of how can we use these novel tools and novel approaches to actually generate tools that we can use for regulatory monitoring purposes. We have a steering group made up of a mix of environmental freshwater organisations who are interested in environmental conservation, regulators, and commercial water companies. And so in terms of the project, we're interested in how you go about understanding how freshwater microbes respond to chemical pressures. And we have a range of different approaches available to us that can sort of broadly be divided up into these 3 categories. And they're on this gradient on the bottom. On the left-hand side, we have highly tractable, highly reproducible experimental approaches. And on the right-hand side, we have approaches that are less tractable and reproducible, but have higher levels of relevance and complexity. So for laboratory-based experiments, highly controlled, high throughput, high reproducibility, but the criticism is they maybe lack complexity and therefore lack realism. In the middle, we have mesocosms, either indoor or outdoors. And in the case of Pacific, we're using outdoor mesocosms. And these sort of have the best and worst of both worlds. So they're sort of semi-controlled conditions. We have medium levels of throughput, reproducibility, complexity, and realism, but they sort of hit a sort of sweet spot in the middle in terms of using these to understand these pressure response relationships. And then on the right-hand side, we have field observations. So the criticism of these is they can be quite uncontrolled, low throughput, because it's a lot of work to go out and collect samples, maybe lower levels of reproducibility, but the advantage that you're looking at real world observations, so high complexity, high levels of realism, and sort of useful for trying to understand what's actually happening, but quite difficult to disentangle the data sometimes. And it's along this gradient we've designed our approach for Pacific. So we have laboratory-based experiments, we have more complex sort of mesocosm-based experiments, both in the lab and in the field, and then we have field-based observations. I'm just very briefly going to talk you through how the different work packages are approaching these. So for work package 2, the intention is to use these controlled, reproducible experimental approaches to understand which type of chemicals should we be focusing on, which ones actually represent a threat to freshwater ecosystems, and then perform more detailed experiments where we can understand these different endpoints, these taxonomic endpoints and these functional endpoints, to understand at what thresholds do these chemicals actually start having an effect. So for this approach, we can really take advantage of the fact that microbes are very small. It's a real, it's an advantage we have over other people doing ecotoxicology with larger organisms. So within a tenth of a gram of sediment, 100 micrograms, is about 10 million cells. So still an incredibly complex and diverse community. And this ability to miniaturize our exposures means we can use these 96 well plates. We can use robotic systems to do lots of exposures in a short space of time. So this approach will enable us to screen a large number of chemicals. We're using oxygen consumption, so the metabolism of these communities, as an endpoint, and we can screen them to identify which chemicals are likely to be having an impact on these communities. And our working hypothesis is that the antimicrobials are likely to be very strong candidates. Once we've identified a subset of these bioactive chemicals based on based on the likelihood of them actually being measured in the environment, the intention is to do more detailed experiments where we use a gram of sediment, so up to 1 billion cells, so highly complex communities, and perform these longer exposures across a range of concentrations and measure these different endpoints. So the community composition, the genetic measures of the function, the metabolism, and these enzyme assays that can measure measures of the functional component of what these microbes are actually doing in ecosystems. So this should give us a better insight into what chemicals are important and at what concentrations might they be having an effect. But the criticism is with lab-based exposures is they lack realism. So to address this, the next stage is actually to move out into the field and perform these mesocosm-based experiments. And this will enable us to look at realistic exposures and understand how multiple stresses might interact to affect microbial communities. And this is work that's led by Michelle Jackson at the University of Oxford, and her team, including Sam, had this fantastic system called the Extreme system. It's effectively a scaffolding setup that can take sets up a series of 25 centimeter continuous flow mesocosms outdoors. So it takes water from a river. In this case, we're sampling both upstream and downstream of the wastewater treatment works. It pulls it into a header tank at the top. We can then split this between an ambient and heated treatment, and then these run through into these 25 centimeter mesocosms. Within these, we've got various different materials for microbes to colonize. So we have 1 centimeter as porcelain tiles, we have stones, we've got cotton strips to look at microbial degradation, we've got leaf packs again to look at microbial degradation. And these get naturally colonized. So we're looking at real complex communities of microbes exposed in an outdoor setting, but with an element of control on the top of it. And these experiments, so Michelle and her team were incredibly efficient, and they ran these last year. These were run at 4 different wastewater treatment works within the Bristol Avon, sampling both upstream and downstream of wastewater treatment works. So you can see here, these are the scaffolding setups. These are the mesocosms just before the 30-day inoculation period, and then they're run for another 30 days to actually allow the temperature treatments to establish. So we have completed these experiments. We have a huge number of samples that are currently being processed in the lab at the moment. No data to show, but I'm really excited to see what comes out of this. So the final sort of part of this gradient along the top is looking at the most realistic and most complex data, which is what we collect from the field. And we've got a number of different aspects to this. So we're sampling in 2 catchments. We have the Bristol Avon, where we've already carried out 2 sampling campaigns. Again, this has generated a lot of samples that are being processed in our labs at the moment. We're planning sampling campaigns in the Thames. And for these catchment-based sampling, we'll be looking at paired genomics measurements, so looking at the composition of microbes and their functions, with high-resolution mass spectrometry, which looks at pollutants in the sample. We also have a fantastic aligned project. So this is funded by the Environment Agency, where through their funding and in collaboration with them, we're looking at a subset of nearly 2,500 biofilm samples that have been collected from across England as part of their river surveillance network. And I'll talk a little bit more about that in a minute. So the final work package is how do we actually use these data to sort of create useful tools that might be helpful for actually managing freshwaters. And we have two modelling approaches that represent the two main pathways that pollutants get into the environment. We have a point source modelling approach, so this is wastewater sources, and this is developing in collaboration with a company called Wallingford Hydro Solutions, who develop a hydrological model that's widely used by the environmental regulators in the UK, a water quality extension that will enable us to look at the fate of chemicals that go through water treatment works and out into the freshwater environment. The second component is a diffuse source modelling approach. So this is using a model called INVEST. This uses UKCEH's crop land cover map to predict agrochemical applications to land and uses hydrological routing to look at the pathways through into freshwaters. And the intention is to sort of bring this all together where we have our field observations from work package 1, we have our experimental thresholds from work packages 2 and 3, to generate catchment risk maps to understand where the likely hotspots that chemicals could be impacting the health of the microbial communities within freshwaters and how that may affect the wider freshwater ecosystem health. There's a final component to this where we actually want to sort of forecast into the future and think, well, what would happen under future change, whether that's climate change, land use change, hydrological change, socioeconomic change, to see whether or not these threats are likely to change. So, for example, you can imagine under a scenario where we have hotter, drier summers, wastewater dilution may get less, and that may have impact. So these are the sort of scenarios we can model. And then the final thing I just want to highlight are some of the data that we're working on at the moment. And as I said, this is a parallel project funded by the Environment Agency, but it has very, very close alignment with Pacific, and we're going to be analysing some of the data under Pacific in collaboration with the Environment Agency. So this is 2,400 samples, I think as previously said, higher than that, that have been collected from across England as part of the River Surveillance Network. We've harmonized the methods that we've used on this project with what we're doing in Pacific. So there's a nice link between what we're doing in the lab, what we're doing in the mesocosms, what we're doing in our catchments, and then what we're doing at national scale using the same methods and approaches. So from these samples, we've sequenced 1,600 samples for different taxonomic markers. So this is for bacteria, for fungi, for micro-eukaryotes, and an assay that looks specifically at diatoms. We've also done metagenomic sequencing on 450 samples. So this is the way that we can look at the functions within these microbial communities. And this has generated an incredibly rich data set that we can start to mine and look to develop useful tools to find a new way really to look at freshwater ecosystem health. So just to give you some quick findings, this is some taxonomic correlations that we've found between microbial markers. So this is just for the bacterial community. What we have here is a heat map on the left-hand side. The red boxes represent positive correlations, the blue represent negative correlations, and the significance is in the box. So what you can see here is there are groups of taxa that have strong positive or negative correlations with these environmental variables. We can also use more sophisticated machine learning and other approaches to mine these data to look for taxa that are related specifically to particular variables. So on the right-hand side, we're looking at nitrate. These are taxa that are either negatively in blue or positively in red correlated with nitrate. Now, just using microbes to predict nitrate isn't particularly useful because we can obviously go out and measure nitrate. But the hope is we can start pulling in more data sets, the agricultural, land use data, the wastewater data, to actually start predicting the likely the stress and the health on these freshwater ecosystems and develop new tools that we can use to diagnose the status of the ecosystem. The other component is we have these functional markers as well. So on the left-hand side, we've got an array of different classes of functional genes. And again, we can look at whether they're positively or negatively correlated with different physicochemical parameters. We can sort of delve into this in more detail. So this is a plot looking on the right-hand side specifically at nitrogen cycling processes and their relation to these physicochemical parameters. And again, there's huge potential with this data to start generating quite sophisticated approaches to measure ecosystem health, but hopefully to start diagnosing what the main drivers are. There is a large suite of genes, the antimicrobial resistance genes, that have evolved specifically to respond to different types of chemical exposure. So the hope is that we can start using these to actually, delve more into the data and actually use this to understand maybe the different sources of pollutants and how they might be impacting on these ecosystems. So summary and next steps. So we are ongoing with the sampling of work package 1. We've already carried out two rounds of sampling in the Bristol Avon, and we will be doing some sampling of the Thames catchment. Laboratory experiments and optimization are ongoing. The extreme mesocosm experiments have been completed, but we have a lot of samples that we're currently processing in the labs. We have this fantastic ongoing collaboration with the Environment Agency to develop new indicators of freshwater ecosystem health. And my hope is this is a really nice pathway to impact from the project, where we can start developing tools that have real potential use in real world regulatory monitoring situations. And I should say, as a final point, really welcome to have new input from a wider range of stakeholders and interested parties. So please do obviously ask questions now or drop me an email. I'll be happy to respond. So thank you for your time.
Thanks, Dan, for a real good summary of, variability in freshwater quality. I like really to hear about the historical context and, an overview of the project and where you've got to. So if anyone has got questions,